Just before we start with that, just a reminder, if you are interested in learning how the GlyGen webpage works, please join the demo session. If you are more interested in that aspect of GlyGen similarity, and how we deal with the fact that some GlyGen structures have missing informations, but they are still related to the other GlyGens with the complete information, please join that session free with Nathan. Just so you know, and as a fair warning, this meeting is supposed to be very interactive, so not much demo, not much slides. We actually hope for a lot of feedback from your side. So, Rasha, do you want to start?
Well, hopefully we can all feel free to unmute and contribute as much as you want. We've all been on enough Zoom and other forms of meetings over the last couple of months that we probably have we know how to behave politely in these sort of massive meetings. So I'll just say a few words then, and then Raja, please jump in. I think this, as I mentioned earlier, this session will be the most useful for GlyGen, and so we appreciate your input. As many of you, as you remember, we had asked when you registered for these meetings to give us some ideas of the kinds of things that you might like to see GlyGen develop that would help you in your research or in your specific questions that you might want to address. So we've kind of compiled some of those that we received as new suggestions here in this use case document on the top, and on the bottom, we can scroll down a little bit, Raja. These are some of the original use case suggestions that we had at the initiation of GlyGen, and some we haven't been able to address yet, obviously, and they might be worth rediscussing, especially number 14 here. It would be nice to have a glycoinformatics resource that doesn't go dead after a couple of years. We think that's important too. And so talking about how we can have sustainability would also be very helpful. So if we scroll back up to the top, Raja, I think we could just, to get things started, we could look at these new use case suggestions that came in. And if anybody suggested these, we could start talking about those and just kind of go from there. Anything you want to add to that, Raja?
It looks like a few people are saying they can hear multiple people speaking. Are you logged into 2 WebEx meetings at the same time? Did you log into any of the other links? Because if you logged into the other link, that's going to cause an issue. If you just want to choose one. So if you chose session C or B, you need to exit this. Just leave this meeting.
Okay, maybe one suggestion or one piece of additional information regarding these use cases. When we are talking about the use cases, we are actually thinking about questions you may want to ask our database that you cannot ask another database because we don't have this data or we can't answer these questions. And these questions can be quite complicated. You know, usually you can say something like, find me all proteins that have this property. You know, UniProt does this, we can do that, no problem. But very often you have questions that go across multiple domains. Find me all proteins that have this and this glycan attached, or this and this type of glycan attached, and at the same time have this and this function, stuff like this. And we are really looking forward, or we are really hoping for you to provide us this kind of questions, because that's what we can implement in our portal. You know, we are not making that portal for ourselves, we are making it for the community. You are drivers on that one, so any feedback you give us is good. And Rasha, do you also have the quick search open? So one way how that can look like in on the portal is we have all the search interfaces, and if you want to learn about them, there is this session B. We will go into detail on that. But we also have that quick searches there, where we essentially take that use cases, this kind of questions that people asked us, and we put them on that page. And if you click on one of this quick search, one form will pop up, which is very simple, with one input, right? And then you get the answer to your question. So really think about any type of questions that you can ask, and you would like to have an information resource that can provide it to you, but none of this exists right now. Give it to us. It doesn't mean we can actually do it, but just by you providing this input, you give us a target to something that we can work towards. Okay, so is there somebody here that would like to start with a question that you have?
This is Laura Mahal. I don't have a question, but I have a comment. And it partly has to do with the quick search. So one of the issues that I found when trying to do like sort of the quick search is the fact that like all of the glycans, for example, here you have to go to the gly2chan nomenclature. In the other, you have to go to the UniProt, and you can't search in the quick search just by a glycan name. And so I'm wondering, in terms of usability by people who are not as familiar with using multiple databases, if there's a way to simplify that for the more general community.
Okay, that certainly is a good comment, and we get that a lot. The main issue we actually have is there's no good, dictionary with all the glycan names and their synonyms. We all know if we talk about MAN5, what this means, but that's just one example. Another alternative that hopefully might also help is we are thinking about integrating graphical input tools. Rasha was talking about that in the morning, where you can not specify a name, but you can draw the glycan rather than having to know this awkward GlyGen accession. So Laura, are you actually aware of any resource where we could just get a bunch of glycan names and their synonyms and the structures?
I don't. I mean, one possibility is to do it by the submotifs, right? So, I mean, you guys are curating motif lists, and one could imagine that you could characterize glycans by that. But I don't know of a single source that sort of has all of the interconnections. And I think that is, I agree. I think it's a big problem, and I think it's a big issue for people coming into the field, right? Although a simple thing, I mean, and this isn't on the glycan side, but on the protein side, is that again, like with the proteins, instead of using sort of the gene names for it, which is sort of what I expected, the protein requires that you actually, again, use the XPASI, which I think is a little bit complicated for people, because I had to go to the, not the XPASI, sorry, the UniProt. So I had to go to UniProt in order to look for, and look up the protein in UniProt in order to be able to input the protein for something.
So actually, Rasha, if you go back to the simple search, you should be able to drop a protein name here. Laura, do you have something on top of your head right now that we could use as an example?
L1CAM. I was looking up L1CAM just because we'd worked on that, and it is glycosylated. So.
Okay, let's see what happens. And if nothing happens, that's certainly something we can look into why nothing happens.
Okay, so this comes up with it. What's interesting is when I went to the thing, because I did it by the, examples where it says look for a protein, and in that it said you had to put the UniProt number. So I guess in the more general one, it doesn't say that, but in your specific examples, when you say I have a question about, it says to put the UniProt number. So that may just be a disconnect between the different places that you can put things in.
I see. You're talking about the quick search. You try to use that in the quick search. Okay.
Yeah, and you can't do it there. So, which, it seems like the quick search is actually less quick than doing it this way.
Okay. Fair enough.
Renee, do you want me to try the same search in the global search?
Sure, go ahead. I mean, but if the simple search brought results, the global search certainly should also bring results. But what Laura said is true. The quick searches are tied to a specific type of input, similar to the advanced search. So maybe we can relax that more and also allow, gene names or protein names instead of just the protein accession.
Yes, Rene, that's what I was gonna say. We could relax that, but there is a potential that we might get empty results if the name is not found. But that's the same thing with the ID also.
Yes.
Laura, you bring up a really good point. People can come to a resource with the name of a protein, whether or not it's a uniprot accession, a name that they're familiar with, and probably get data. But for glycans, there are no nice names like that. And even motifs can be a bit of a challenge to people that don't understand what a substructure within a glycan is and the history of how it got named the way it got named. And so it may be that we don't have a way to allow users to come to the glycans easily, and that they have to go through the proteins or through the functions or through the diseases or through the expression patterns or something. There's not a language for describing glycans that's easy.
So I'm just going to follow through with this search, Lara. So in this particular case, I'm just going to click on the gene row, and so let's see what we get. So it does get the protein. If I were to click on another row, we might get a slightly different result because it looks for the presence of that. But I agree. Making it consistent across different types of searches is something we need to look out for and see how best we can do that. So the question that Lara brought up, Mike, from the text mining, as we are pulling up some of these names and we are creating the first draft of the dictionary from how people are using it in publication, that would be the step one, right? From there, one still would need to map it to structures and motifs. So that's the challenge. And a lot of that has to be done at some level manually by experts and or at least using some rules. Some of that Ethan most probably is talking about right now. Is that how it might help in the future? Not yet, but in the future?
One thing I was thinking about is another option is if we cannot retrieve these names and synonyms from somewhere, we could also think about adding an option on the GlyGen page where people can actually go in and propose a name. If nobody has the list, we could create the tool which allows people to curate it and then just assume that over time this synonym list will grow and obviously it gets immediately integrated in the search. It's something to think about. It obviously needs community input there because somebody has to go in there and actually make use of the tool, but it would be a solution. I saw in the chat there is a question from Scott Miller about the allowing to search for in pathways. Rasha, do you want to address this?
I'm looking for the question right now. Let's see.
Okay, so he was saying the earlier presentation made references to biosynthesis. Are you envisioning a searchable database for pathways? A list of enzymes associated with a particular structure is nice, but pathway searching would be cool and useful.
Rene, so So I think we can associate those enzymes with the pathways they are involved in, and then we can search through those pathways. I think that should be easily extend. We can extend that to pathway entries.
So Scott, I'm not sure.
Yeah, I can say a little more when I'm thinking. I don't know how universal the interest in this would be, but I can imagine that in thinking about biosynthetic pathways, if you were able to identify sort of nodal relationships across intermediates in a pathway, you might be able to begin to speculate about evolution and then ultimately correlate that to functions. And that data is probably collectible, mineable, but maybe not necessarily extractable from a database that is sort of binary enzyme to biosynthetic step in a biosynthetic pathway. So I don't think this is trivial, but I'm sort of recalling that things like this exist in other parts of metabolism. Probably others on the call will know more about it than I do, but I would think it would be, just intuitively, it would be quite cool if you could adapt that for glycobiology as well.
You're absolutely right. And Will York is working on that now, as several other groups are working on how to map. It gets combinatorially complex as you get branches and different linkage options, but it's a very useful way to, I think about diseases that affect glycan biosynthesis and ways to link together various kinds of information. I think it's a worthwhile goal that we need to pursue and are.
I just jumped in. This is Laura. And we could start with microbes because it's much simpler and everything is connected. I know that that's not been a priority for this, but it's a little bit hard because that's, I think, where you could get some real traction in linking the pathways to what glycan is produced. Anyway, it would be great if there was some parallel effort that intersected with you guys, because I think it would be so much easier to make progress on that issue.
So Renee, can you go to one of those enzyme proteins entries through this biosynthesis quick search and go to the enzyme page and go to the pathway?
Actually, Rasha has the control. Rasha, can you just follow Rube's hint?
Quick search, just hit quick search.
So I'm just going to respond to Laura's comment, and I'm going to go there, Ravel. So this is our, and yeah, I mean, I do agree with you. Getting the microbes in is going to be really important. And the only thing, what is happening is that anytime we bring in a new group of organisms, we have to make a lot of decisions. We have to kind of think forward and see, will this break something? Is it going to be able to handle, the data model is going to handle or not? And every time we bring something in, we brought the viruses in. And as I mentioned in my talk, I mean, it was great that we brought the viruses in. We could get the SARS-CoV in as soon as the pandemic started, pretty much. So for the bacteria, for microbes, we would like to bring in, but first we looked at the humans, and then we had to bring in the orthologs for the humans, or the model organisms related to the humans, because then you can do certain types of searches. So bringing in human was hard, and then when we added mouse, it was easier, rat was even easier, and so on. Same way when we added HCV was hard because we had to make some decisions, and these decisions requires a lot of back and forth between experts in the field and ourselves within the GlyGen team. So we would love to bring in bacteria. It's just that we haven't come to a point where we know when it will be. It will be there in GlyGen. I mean, I'm confident of that, but it's just we do not have a plan yet exactly when it will be. But we'll continue talking internally. We have our virtual face-to-face meeting this summer where we are trying to figure out what are the next set of organisms we'll bring or kingdoms we'll bring in. So we will have a better idea about when that will be. Now, the question that might have come up is that is there a concept of soft integration, which means we really do not spend as much time. Is there some way we can still do it without going through all of the processes? And there are pros and cons to that. We have to think those through before we do that. But I do hear you, and it's something that is top of our mind.
So Laura, this is where we need experts to advise us too, microbial glycobiologists. I could see how one could think about linking through host binding protein specificities to subsets of microbes that then one might want to expand in terms of knowing their biosynthetic capabilities. But how rapidly would that just explode? And if you had, say, a 5 mammalian binding proteins that recognized, how many microbes would it be? Thousands? Or is there's something more limited that one could think about starting with?
Yeah, we're not, I don't know that we know that answer yet because we're just kind of at the infancy. But, I guess I was thinking, there's a bunch of Kleb strains, and we know that, whether they have capsule or not matters. And the same with Ruminococcus navis. So there are some strains that you could go in where we know a lot about what glycans they make, and we have the genetic, they're all sequenced. And that could be a good training set to start to bring these things in. And those are, Klebsiella pneumonia. We could start with the escape pathogens because they're so important, and their coat varies, and that can influence their pathogenicity. So, I mean, Sean probably has good ideas as well, but, I was just thinking like Pseudomonas aeruginosa, Klebsiella pneumonia, it's not, we wouldn't have to have everything right away. Right. And the Ruminococcus navis strains are associated with like inflammatory bowel disease, different ones with either health or non-health. And so again, there's something known about the biosynthesis. So if you pick some things that were already associated with the disease, it would just be useful and maybe a place to build from.
Right. You could interlink several different types of data that we already support. Yep.
Right. That would be, it would be really transformative to be able to go back and forth from host to microbe or host to virus. But I want to say that I'm really excited about where the database is. I feel like you guys have made a big jump in the intervening year, like from last year to this year. So I just want to say awesome. I'm excited.
Thank you. So Mike, maybe we can continue this discussion. As I said, it will be there. It's just that we need to figure out when it will be there, and then is there any opportunity to provide some type of softer integration? I do not know what the answer to that is, but we definitely will look at it and try to come up with some kind of a plan when something like this will be could be done. So regarding the pathway question that Robel, you wanted me to try, can you please tell me where do you want me to go?
Quick search, Raja, and then go to that biosynthetic, the first one. and then just use that glycan and search. So take any one of the results and then go to the pathway section of the proteins. So basically, we could have searched the same quick search by using the pathway identifier instead of with the what I'm trying to say is, no, I think it's the other way. So if you want to say what enzymes, what are the enzymes that make this glycan, it could be what are the pathways by which this glycan was made. So we can search the by pathway, glycans by pathway, by connecting, enzyme to glycan. Then we have a connection from pathway to enzyme, so we can jump from pathway to glycan. But I don't know if that is going to be a correct. We are assuming that if this enzyme is involved in this pathway, then that pathway is, and if the enzyme is making that glycan, then the pathway must be making that glycan.
Yeah, we'll figure it out. I mean, these pathways branch. They're not simple, straightforward metabolic pathways that start at A and go to B, but actually one glycan can show up in several pathways, and one enzyme can show up in several pathways towards a different glycan. So it'll be a challenge, but we'll get there. What we have now is displays current knowledge, but I think there are ways that we can improve that. So are there any other sort of use cases that you'd like to share? We'd love to have some more input, and also we'd also like to share, if anybody's interested, strategies for sharing data, but we'd certainly like to collect any additional ideas that you have about interesting uses. Questions you'd like to answer? Well, why don't we take 5 or 10 minutes and talk about data, Raja?
Yes. So I want to share some slides. Let's see. So at this point, when we're talking about sharing your data, we are mostly talking about data that you have in a table. So the table can have, each of the columns can have information. And once you upload it, so if you go to data, and I'm going to show some examples of how the tables look like. So if you go to data.glygen.org/upload, you also have a link right on the top level menu, and I'll show you how you can get to that. You will be directed to a page where you are prompted to choose a file. And if you want to see how an example file looks like, you can click on the example files in CSV, which is comma separated, or TSV, tab separated. So once you submit file, there will be some automated QC checks to ensure data integrity. And these QC checks, it is a continuous process. As we get more and more data, we realize that maybe we should do some different type of QC or add something, or sometimes we want to refine an existing QC check. Some of this I mentioned in the main talk. So once you upload the file, You choose the file format and select upload. And then there is some basic sanity check. The results will show how many rows, how many columns, and if there are any failed rows. A failed row can happen, let's say if something is missing a comma. At least here you will be able to see what you were expecting from the upload. In this example, we have no failed rows that need review, so one can proceed to the next step. So for mapping to GlyGen IDs, so we don't, GlyGen doesn't really have its own IDs, at least outward facing IDs. We are using existing ID and accession space that is commonly used. So for proteins, we are using UniProt accessions. For glycans, we are using glycocan, but we can also handle some other type of IDs and accessions which are also very, very popular, such as RefSeq or PubChem or KEBI. So some of them are still not integrated into the upload system, but conceptually we can provide this type of, we can provide what is called ID mapping. So you can actually have a set of ID and then you can map it to another set of IDs. So then here you select the GlyGen ID type from user ID field from the drop-down menu, and then you click submit. So for this example, we have chosen the GlyGen ID type is protein and user ID field name is UniProt KB accession. So what the next step is, we can do a protein site check, evaluate the uploaded residue information for specific positions in the protein. The protein site check is select from the drop-down menus, protein ID field, protein position field, and user protein residue field, and you can then click submit. So once you do that, then you can also, by the way, you can also have additional columns. So these are the main columns. These are the primary columns pretty much, which will help anchor the table with any data that we have inside of GlyGen. I'll give you an example. So for example, UniProt has Tremble accessions which were submitted. So Tremble, because somebody submitted a gene sequence and it has not really gone through the process, and it is not mapped to a canonical protein sequence in the human proteome. So this Tremble accession most probably either is a fragment of an existing protein that is already there in the human proteome list, or is an isoform. Most of the time, though, it ends up being a fragment because the full-length protein or the genes was not submitted. So if you were using such an accession, it will not map to our master list and it will throw an error. So in these kind of scenarios, we are thinking of providing mechanisms such as BLAST or some other sequence alignment tools to identify what is the correct accession of your protein that we have in GlyGen, which is the reviewed, approved through collaboration with UniProt. We maintain a regular list. So this slide is about records preview. With records preview, you can check that the uploaded data is previewed correctly. The report will list all the fields and data from uploaded file. And in this particular case, it gives you the UniProt accession, it gives you the glycosylation position, it gives you the residue, and so on. This is a way of checking to see if it is matching what you are expecting to see in your data set. So here you can save the file. When the file has been uploaded and passed all the QC checks, it can be saved in GlyGen, then fill out the information fields, first name, last name, email address, institution, and click save. So this is the very basic thing. So this allows some of the initial checking to be done at the user's end, which also allows us to then contact the user and clarify anything that is, let's say, missing, or for some reason, let's say the user has a table which has different types of accessions which we do not support. This also gives us some feedback maybe we need to support some additional type of IDs or accessions. This overall is really important. It helps us to increase the coverage of glycosylation sites that GlyGen provides. And in terms of the organisms we have, we are collaborating with all these resources that you all are familiar with, we're familiar with, to make sure that we are sharing data amongst each other. So using this approach, getting data directly from users, which has already been published, and getting data from existing resources, it will help us continuously maintain a comprehensive data set.
I think
and a little bit of additional time, if that's okay, to go to the GlyGen website. So
I think, Roger, you've got 5 minutes. We'll go to 5:05, and then we'll have to switch sessions.
okay, so I'm going to go to GlyGen.org. It will take me less than 5 minutes. I just want to show how to get to this page. So this is our home page. If you click on data, and then if you go to the right, there is a place which says upload file, and you will end up on this page. So that's it. And depending on how much usage we get from the community and how much data we are collecting, we do plan to expand this to also upload other types of data. And if you look at how the data looks like when it is available, your data, this is on data.glygen.org. So this data is not integrated. Well, they are integrated, but your data will not immediately get integrated. It will go into the next cycle. So this data, let's say somebody submitted human glycosyl transferases. So you can see how it looks like. It has a little bit of a paragraph which explains how it was created. You will have example. Actually, we can show an example which was submitted. Maybe Christina is on the call. Hopefully that search should bring it up. Yeah. So this was submitted. In this case, we did not have the interface available, but we did interact via email, and now we have an interface. It will be a little bit easier. So what she submitted, we worked with her, of course, session numbers, glycosylation site, in unipart KB, amino acid, saccharide, glycosylation type, cross-reference key and composition. Some of these columns are automatically generated, but most of them were submitted. And if you want to read how we cite the work, it says authors Christina Wu, and so on and so forth.
Okay, I think what Raja just showed is very important also for everybody in the chat right now, because one of the biggest complaints we always get is that people look up their favorite protein and then there's missing information. Like the GlyGen we just published is not in our database. And the reason for that is that in the past, GlyGen was a data integration effort where we integrated data from other databases. But there is so much information published that doesn't make it into these databases. So we are missing out on a lot of this stuff. What Raja just showed is a way around that where you publish your data set. Very often you have supplementary tables which pretty much look like what he just showed us. So in addition to publishing that information, you can also upload it to the system, share it with the community in that way, and then it will be available in our database and also help others by just having more information on the site. And as he showed you, we will reference you that this data came from you. So please, everybody, just think about what kind of data you're producing, what kind of data you have available, and if you are willing to share it. And, just contact us, and we are very happy to work with you on making that happen and integrating these data sets.
So I'm done. I'm just kind of quickly showing how that data ended up in our front end. So this data was submitted by Krishna Wu, and it's in the front end. Not so the table feeds into a triple store, and that feeds into the front end, and of course the APIs and so on.
Well, Amanda would like us to give everybody 5 minutes to log out and log into another session if you'd like to. But thank you for contributing and participating. And if you think of burning use cases in the middle of the night, please feel free to send them to us. Your input makes us better.
Thanks, everyone. I put the links to the other sessions in the chat again. And yeah, we're going to start the next session again at 5:10 to give everyone a chance to switch. So feel free to log out and log into those, and then we'll all meet back here just after 6. Back here, by back here, I mean in the same room. Mike and Raja, do you want me to leave this as one large recording or make 2 separate ones?
Well, I think one large is fine.
Okay, I'll keep it running.
Less
Thanks.
things to keep track of.
Anyone that's just joining from one of the other sessions, we will start at 5:10. We're just allowing people to switch over from one session to the other right now. Thanks. Okay, it looks like we had some people switching around. Some people are still here, so but I think we can get started again.
Okay. Thanks for sticking around if you're still here. We'd love to have more comments from everybody, and thanks for joining us if you're coming from another session. I hope the other sessions were useful to you. So this, I think, might be the most useful session from the standpoint of GlyGen. We're really looking for input from you. We're looking for this to be an interactive 45 minutes or so, where you can tell us how you think GlyGen can help you. And I maybe start by letting Renee describe a little bit about use cases and how we've used them in the past and what we're looking for now in terms of use cases. Renee?
Okay. When we say use cases, what we mean with this is actually a question that you have in your mind that you want to answer, but you cannot go to any database, existing database right now to get the answer there. Either because we don't have the data or because it's a multi-domain question where you want to ask questions like find me all proteins that have a certain function and at the same time have a specific GlyGen or a specific GlyGen motif attached to them. So UniProt can't do it because it doesn't have the GlyGen part. Usually GlyGen databases cannot do that because we don't have the protein function part, but GlyGen, for example, can do that. But it's a relatively complicated question, and even in GlyGen, to get the answer to that might be challenging. But what we can do if you have this kind of questions, we have an easy way of integrating that question. Amanda, could you give the ball back to Raja? Or if he already
has it.
Okay. So Raja, if you can go back to the quick search again for a second. So when you're going to the GlyGen web page, we have in the menu on the top that quick search entry, and these are exactly these type of use cases and questions I'm talking about. And the way how you can use them is, send them to us, and if we cannot easily do that with the existing search, we might add that use case here as an additional question, where you can, when you click on one of these questions, it opens a very simple form field where it usually asks just one piece of information. Like for this question here, what are the enzymes involved in the biosynthesis of GlyGen X? Give us the GlyGen, and you will get the answer, what are the enzymes. What we really would like to have from you is this type of questions that might help you in your research. So if anybody is here, just feel free to unmute yourself and speak with us.
Raja, can you pull back up the spreadsheet of use cases?
Right.
Or not?
Right.
Great. Some of you provided use cases when you registered, and these are summarized here, and we can, if any of you want to elaborate on those suggestions you provided or have others, that would be very helpful. We have many use cases from the original design of GlyGen in the planning year. We haven't been able to meet all those, and including this really wonderful suggestion that it would be nice to have a glycoinformatics resource that doesn't go dead after a couple of years. So I think we can all agree on that, and the sustainability is really important. So there are additional use cases that we haven't been able to get around to addressing yet, but we would appreciate your input.
So if somebody wants to step forward, just again unmute yourself, or I already saw there is a question in the chat box. Okay, so one of the questions would be, which glycoproteins are known to be sialylated with ST6Gal1? I'm just trying to think if you can answer that on the front end right now. I don't think there's an answer on the front end. So that would be a potential candidate for a new use case.
Nora, I appreciate your interest in ST6-Gal1, but if I can generalize it, the use case there would be what are the protein substrates that are associated with specific glycosyl transferase activities?
Right. So the type of enzyme or the instance of the enzyme would be something the user could provide, and then he could get all the glycoproteins where that enzyme may have worked on.
GlyGen.
Or, well, she was asking for the glycoproteins, so from that point. Okay, thank you for that input. I think you cannot do that easily on the front end right now. The data is actually there, although we are improving the enzyme glycan relationships right now. But that might be a use case we can add in the future.
The tricky thing there would be, for instance, Nora, to distinguish between ST6-Gal1 and ST6-Gal2 substrates, for instance.
Okay.
That might require some knowledge we don't know yet, but we could also indicate ambiguity when we don't know which particular silotransferase may be acting.
Okay. There was a second question. Have you included different sialic acid forms and their occurrence in different species in your database? So on top of my head, my answer would be we have new GC and new AC, but all the different variations of, additional acetylations and methylations and stuff like this, we hardly have any glycan structures simply because we are not really present in the other databases. So that would be a question for GlyGen. Do you have this kind of information? So something we could actually get from you and integrate in our data resource, or are you way? Yes, there are publications. The problem is just extracting this information from the publications is not an easy feat. And as Rasha was talking about in the morning, we have a data mining, a literature mining effort going on. But that actually focuses on finding proteins and their sites. Extracting the glycan structures out of papers is much more complicated. And then if we actually talk about different sialic acid forms, that's just one step further. It's not impossible, but right now we are limited by what we have available. If somebody actually working in that field would provide us information in some kind of form, like a spreadsheet or so, that would really help. Yes. So if somebody wants to volunteer to collaborate with us, please, just shout or send us an email. We are very integrated. We are very interested in working with you and getting this type of data. Actually, Rasha, since we are already on that topic, maybe you want to give a short overview about that data submission system, because that ties right into that question. So if not just this sialic acid question, but if somebody of you has data available, glycan data, glycoprotein data, glycans attached to proteins and specific sites, this kind of information that came out of the work of your lab, we have a system where you can actually submit that data to us, and then we can share it via the GlyGen platform with the community. and just provide more information, make that system more useful. And of course, we will acknowledge you. Raja?
All right, thank you. Actually, it's great. Shichen, your suggestion is exactly what I was hoping would come in. So as we mentioned earlier, we are limited with what information was already available in databases before we started. And what we did was that we actually harmonized existing data in existing resources, QA, QC them, standardize them, and so on. So we have started this GlySpace Alliance, where we are working with the major glycoinformatics resources, GlyCosmos and GlyConnect. And Unicorp KB is already part of GlyGen, or they support, they're a sub-awardee of GlyGen. So we are working with at least some of the major resources. Now, what was missing is individual investigators, like you mentioned. So individual investigators, they have publications, they have supplementary tables that they're submitting in the publication, with the publication. And these tables are important. They're rich with information, but one of the challenges is the table, they do need to have certain types of accessions and IDs. What do I mean by that? So what I mean by that is that the table, let's say a user wants to submit. I'm going to use the example of protein at this point, just to show an example. So if an investigator has a table of glycosylation sites mapped to UniProt accessions, what they can do, they can come here, and also glycosylation structures. They can come here and they can upload. So this is data.glygen.org upload. And if there is, you can have a comma separated file or a tab separated file. And then you can choose the file and submit. What it will do actually, it will do some automated QC checks to ensure data integrity. A lot of things could have happened. Let's say the table was generated 2 years ago, and since then some of the accession numbers are retired. So one would need to map them to new accession numbers. And all of this is important to maintain a resource which is highly interconnected, because as soon as the data gets integrated, you can then see the pathways, for example, the proteins are involved in, the disease information that is coming in, or the GWAS study that was also associated with the genome and the genes and so on. So it becomes, even though a 2 or 3 column table is uploaded, once the data gets completely integrated, the value of the final output that one can see in the front end is huge. So the next step would be, just a second, is you choose the file format and you select upload. And what the next step is first the QC step is called the basic sanity check. It just tells you, the user, how many rows, how many columns, how many failed rows. Let's say in some row, there were no commas. So our row, there was an empty row in between. So it will tell you how many rows were processed. And then in this example, we have no failed rows that need review. So one can proceed to the next step. So the next step in this case is mapping to GlyGen IDs. Now, GlyGen, they do not have their, we're not making up any IDs of our own. We are using existing ID space and accession space. And that's important because it allows us then to easily also map data to other resources like NCBI, EBI, Swiss Institute of Bioinformatics, and other places in the world. So in this case, you select the GlyGen ID type, the user ID field, and then there are drop-down menus, and then you click submit in the user ID field. This is UniProt KB _ accession is the user ID field name. And in this example, we have chosen protein and UniProt KB accession. So once the user, they do this, then they have a protein site check. So the protein site check will actually check and see, let's say for handling glycosylation in the site that is glycosylated, is there aspargine, or in the sequon in the third position, is there a serine or threonine or any of the other non-usual amino acids, but nonetheless are known to be glycosylated. So these are all part of the rules. But if there is an amino acid which is completely not expected in a sequon, then it will throw an error. And the errors will be in log files, which then the user can check and say, oh, okay, this was a mistake, or actually this row, yeah, we should not really submit this row to GlyGen. So let's say there's a proline in the second position. So you would say, no, this is showing a warning. And so there are the concepts of warning and errors like any other thing. So warning is still will allow you to submit, errors will ask you to fix things before it can be submitted. And we are reviewing the quality checks constantly and through feedback, and we hope to improve them as we go along. So this is the records review. So in this case, it will give you on the left at the bottom the UniProt KB accessions, glycosylation position, and the residue. Again, this will give you an idea about how the file was processed, was it processed correctly, and so on. So the last step would be save the file. So when the file has been uploaded and passed all the QC checks, it can be saved in GlyGen, and then you will be prompted to fill out some information. First name, last name, email address, institution, and then save. We need all of this information because then somebody at GlyGen will evaluate what was submitted. There might be a little bit back and forth still because this is not completely automated, as you can understand. We are not there yet. I think for the next several months or maybe a year or so, we will use a system where there is some interaction that is going on just to make sure that it is working correctly. Once we have all of this information in the next release cycle, which can be in a month or two, whichever rows and columns pass all of the QCs and we have an agreement, there is also a README that is created along with this, and I'll show you how it looks like, and it can be shown in the front end. So now suddenly, when there was no data, let's say for a particular protein, we have this new data that was submitted by a submitter, and we strongly encourage people to reach out to us to do these type of data submission, because that's the only way we can make things comprehensive. And I'm going to spend a little bit more time to show how to get to this stage. So if you go to the top level menu, you would click on data, and you will end up on this page. And here you will click on upload file on the right hand side, right hand top corner, and you end up on the upload page where you can do the steps to upload your file. Now, I also want to show you, so I want to show an example of a submission that was done by an investigator. So by Christina Wu. So I'm going to type in Christina in the search box. So these are data tables, by the way. So this page you're seeing is similar to like an Amazon page, I would say. So these are different products, let's say. These products are, most of them are completely integrated into the front end, the demos that you may have seen, but some of the products are still waiting to be integrated or will be integrated in the future. So your file that you submit will end up on data.glygen.org first. It will still be publicly available, by the way, and it will still have a stable URL which you can cite in your paper or anywhere else that you want to. And I'll give you an example. So here I typed in Christina and I do a search and I see this table. So this table, I can click on view details. And if you look at the URL, it is a stable URL, data.glygen.org GLYDS0000142. Now, you will also see there are multiple versions of this table. So the multiple versions of this table sometimes happen because maybe there was an update, maybe the protein accession changed. So there are reasons why there are multiple versions, and you can go to an older version also if you wanted to. So in this table, most of the columns were submitted by Christina's group. There were some columns that we generated, but nonetheless with interaction with her group. So here you see there is an accession number, there is a glycosylation site, there is an amino acid, and then the saccharide, glycosylation type, cross-reference, and cross-reference key and composition. So this type of information, along with a little blurb about how the data set was generated or provided, there are some PubMed IDs, provides all of the related information to this table. Then we also want to make sure that we do the citation. So if you go to the README file, it tells us who the author is, and that's how we keep track. This same information, for those of you who are interested, also is in JSON format, which is also machine readable. And it tells you who the people from GlyGen, in this case, there are people from GlyGen who also interacted with Christina to create this final data set. So we maintain all of the citations, and this is really important. Later on, when somebody publishes, they can go back and see what exactly was done. So we kind of are trying to get rid of this email attachment concept. And this is important for a stability of a resource and also tracing back where the information is coming from. One of the challenges to create this table most of the time will be if the user has a different ID than what GlyGen is requesting. This getting brought. What if you have RefSeq accession? We are looking into providing ID mapping service, which will allow people to create the accession mapping as and when needed. Again, most of the time it will work, but let's say there is a RefSeq accession which does not have an equivalent UniProt accession, those would need to be resolved. But still, it is good to do these kind of resolutions, because overall then in the end, when I go and see, let's say I'm going to go and take a look at this accession here. So if I look at this accession, I will end up with results where this accession is present. I'm going to click on identifiers, and here is my protein, and this protein. If I look at glycosylation, and you see here that this is the position, this is the paper that is associated with it, and so on. So this protein did not have site-specific information. It could have been just in a paper, but because the investigator did collaborate with us and we could collaborate with the investigator, this information now is available and is present in the search space. And one can also look if there are additional isoforms of this protein or not. What are the other pieces of information we have? This is an uncharacterized protein, so there's not a lot of information, but if there was more information, then we would be able to show that. So okay. I think that is what I have to show in terms of data upload. If there are any questions.
Okay, so that might have looked a little bit scary, but you're not alone. If you really want to share data with us, please contact us. We are interested and also willing to help you, setting up that infrastructure to get the right tables in the right table format. So don't be scared. Just think about what kind of data you have in your lab, what you are producing, and that you would like to see and share with GlyGen, and contact us, and we find a way to integrate that data. So we had several comments while we were.
Before we move away from providing data, we're also very interested in pure glycomic data. So we've focused a lot on sites, on proteins, but we also need to build up our GlyCan database as well. So if you have glycomic data that you've performed on a particular tissue or a particular cell type. That's also very useful data for us.
Okay, good. I will just read the questions that we had in the chat, and maybe Raja and Mike, you can address them. So from Xisheng, we had another question. Do you plan to include synthetic glycopeptides and glycans with NMR and HRMS data in your database?
Well, we're already including synthetic glycans. Jan said that we're including many of his glycans, slowly accumulating them in our database. Synthetic glycopeptides, we haven't discussed if that's something that would be of use, and not very specific to one particular user. That's something we certainly could discuss.
Okay.
And links to supporting data is an interesting question. Housing all of the NMR spectra supporting a particular glycan structure is not something we've discussed. I think, Roger, correct me if I'm wrong, but that data we expect will exist somewhere that we can link to, but not that we would necessarily house that volume of data.
So at least currently, yeah, we do not have plans on housing that data, but if there is information on that data and it can be mapped to a particular accession, then we can definitely cross-reference it. So if anybody has suggestions in that area, please let us know. In terms of glycopeptide data, I know Nathan had investigated some of that to see where that can be linked, and we would definitely try to see if we can cross-reference it so that users can then explore it.
Okay. So Nora had a question, is it possible to also include the gene names in addition to the UniProt designations? Maybe Raja, you want to go ahead with that?
It's very much related to Laura's question from the first session about being able to put things in that people are more familiar with.
So I'm just going here, and actually let me start something in the global search. So if I type in SHBG, for example, which is a gene name, and click on search, you will get several results on the protein. I'm going to click on gene, and you can see that you get 3 rows. These are orthologs from human, rat, and mouse, and you can get it. So the answer is yes. Gene name we do support. I don't remember if we are already supporting all of the synonyms, not just the HGNC gene names, but if not, that's something we plan to do in the most probably in the next release or so. It should be pretty easy for users to use gene names to get to the proteins that they are looking for. There is. Go ahead. Did somebody have a question on that?
Go ahead. Finish that. I was going to the next question.
Okay, please go to the next question.
Okay, sorry. We had a question, I think it's actually for me in that case, about the timeline for integrating glycan array data for that. As you heard in the morning from Mike and Raja, in parallel to the GlyGen portal, we are also working about a data submission system for glycan array data. Once that submission system is working and running, obviously we are going to integrate that data in GlyGen as well, so that you can search, or when you're going to a protein, you might actually get a list of glycans that that protein has reported to be binding to. Or if you're on a GlyGen page, you may get a list of proteins that have been reported to bind to that glycan based on the experimental data. We have been focusing very much, or a lot of resources, on that portal in the first 3 years actually, so that glycan array repository was a little bit backlogged on that one, but we still made some good progress on there. Right now we finished part of the submission system where you can upload your slide layout, the glycans and the linkers that you have on your slide, and we started with the metadata and also uploading the experimental data. We are also working together with 10 Faces group and Rick Cummings group, who will provide their existing data sets to us in that repository as the initial seed. So coming back to the timeline question, my current estimation would be that probably by the end of the year, we will be having a first version of the portal running with the data from, or a first set of data from Rick and Ten's group, which then from that point on forward, we can actually establish a pipeline to get that data into GlyGen. So early next year, you might see the first binding information in GlyGen. If you are interested in submitting your own data, that data repository is going to be publicly accessible and everybody can create an account there and submit the data. So it's not limited to our project or to Rick's and Ten's group. We are just the ones that, our collaborators to set them up. Okay, Saraya was asking that first question and just added another question. Will this include the the existing CFG datasets? That's exactly what Rick's group right now is working on. We are working on getting the old CFG dataset. Then Rick's group, after the CFG ended, Rick's group was producing a lot of NCFG data, which is nowhere present right now. And these are the two data sets that his group is working on making accessible to us. So hopefully part of the CFG data set and then a lot of the NCFG data will be the initial data set that will be present in that database. Okay. Nora had another question. I'm not sure if I understood you right. Maybe if you just unmute yourself. So you were asking about to include some information on what is visible on the different tabs. Can you maybe unmute yourself and say a little bit more to that? We cannot hear you in case you are speaking.
Maybe I can try to take a stab at it and see if this is what. So on the top level, there is Home, Explore, Quick Search, Try Me, Data, Help, and more. So it looks like when somebody clicks on Data, you end up with a bunch of tables, and it can be confusing. So why do I have tables here? So one thing that we can do, we can provide once people end up on the domain data.glygen.org, we provide a couple of sentences explaining what one can do from that space, or provide a help link which can go to our wiki page, which explains how to use that. So I think as in this year, we do plan to write a lot of these frequently asked questions, tutorial type of things, and help documentation to allow users to quickly understand when they're in a page, they'll be, I do not know at this point what it will be, let's say a question mark or whatever. So what can I do on this page? Some examples. So we could try to do that and see if that makes things a little bit easier to understand. But conceptually, the data page are just a bunch of tables with different versions and all of that one can download and use. It also is a place, if you're a user and you submitted a table to us, that's a place where it stays. You can go back to it, you can cite it, you can show it when you're giving a talk on how you are collaborating with us. We would love that. If you're collaborating with us, mention that in your talks. So that's what that data space is. All of the data gets integrated into a database, which then feeds the front end, like the search that I just did. When I type in SHBG, it pulls up these entries. So because all of the data is then integrated, they are not lying in silos. Hopefully that answered your question.
I think that's a good idea, Raja, that we have a sentence or two once you land on the data domain to tell people where they are. Because I can see how if you want to get to data, you want some answers, you're going to click on data. And we may need to just educate a little bit.
Okay. One question that, Raja shortly touched on is citations. I actually forgot to bring that up in the last group as well. One thing that you please do is, if you are using our database for something, it would be really good if you also cite us. In biology, if you do experiments, it's quite common in the introduction or in the method and materials, you have a lot of citations where you cite methods and stuff like this. But one thing that gets very much neglected is to reference the software tools and databases that have been used. And we would really appreciate, there is a GlyGen paper out. If you're using our database, to look something up, to confirm something, or download data sets and work with them, please cite our papers, because that's really important for us in the end. Also, if we are looking for new funding, alternative funding, we also will be evaluated by the impact of our papers. And Mike brought in the beginning up that question of sustainability. That's another way how you can really help us to, give us some leverage to get a new funding in the future. Okay. Aria, I'm not sure how much time we have. I think, Mike, you said we start back at 6.
I think Amanda's hoping to get people back at 6 with a little bit of a break before then. So
Okay.
if there are other suggestions or questions, we're happy to stay around and talk a little bit longer if you would like to. But thank you for your input. We're certainly going to consider everything that we've heard today and use that to try and make GlyGen more useful for everybody. If you think of other things, don't hesitate to drop us a note or give us a call. We're happy to talk about new opportunities, new data, and doing what we can.
Yeah, feel free to keep the conversation going, because other people are going to come back in at 6, so we really have 10 more minutes. People will just come back from the other sessions, and then we'll just wrap things up for the day. So feel free to continue talking.
I mean, I can do some searches to while we wait. Why don't I do that for those of you who are here? These are indeed very difficult times, I'm going to pick something that is on everybody's mind. I'm going to pick SARS. So I'm going to share my screen. And I am going to click on, or I'm going to type in SARS. And if I search for SARS, I get several hits. I'm going to go ahead and click on species. And I can see, so when we, in GlyGen, what we do is that we just don't bring in the glycoprotein, because the glycoprotein, they don't exist on their own. There are other proteins within the organism which are also important when people are designing or developing hypotheses or designing experiments. So here in this case, we have brought into GlyGen not just SARS-CoV-2, but also SARS-CoV-1, because one would need to look at these two SARS viruses to understand what's going on. And a lot of work you're already seeing in preprints and right now peer-reviewed publications. So here, I'm looking at, let's say, the spike protein. So I'm going to go and click on this accession, the spike protein from SARS-CoV-2. So here we see that we have links to UniProt KB accession, and so on. And initially, this was called 2019 NCOV. In terms of function, we have some information related to some information related to function, and then we can click on glycosylation, and we see all the sites. So if you look at all these sites, there are several sites on which we have data. There are many so composition-based information. This was curated by our collaborator at Unicarp KB in collaboration with us. And if you look at the entries, initially we just got the information from the preprint. So when the preprint, of course, in this case or in other case, when there is already a publication in the journal, we will update it to the publication. And a DOI search would result in the paper. I'll wait till it loads. One can also click on a particular site to see what are the different compositions that have been observed on that site. So in this case, we are just looking at 17, position 17, and you will see that there are several compositions. We also see something from UniProt KB. They don't provide the GlyCan information, but they do have some rule-based annotation. They had also said this is a glycosylation site based on most probably SARS-CoV-1, if you ask me. And there are 31 rows, which tells you the number of different compositions that have been observed on that particular site. Now, if you go to the sequence view, you actually can go to another site. You can look at the 323 or 325, the O-link sites, and so on. So if you want to explore a glycoprotein, in GlyGen, you can really explore the variety and what is already known for that particular protein and the sites. I'm going to talk about a few things which are we are looking into. We don't have them yet. We are thinking of, even though people are talking about mutations in SARS and how it affects binding and so on. Once some of that information is better resolved, there is a possibility we can provide that type of information also from a mutation tab. But again, that information first has to be validated at some level, published in peer-reviewed journals and so on. So this is how one can explore the different resources. We also have, right now we are also working on developing, it's very, very early stages. By the way, I'm just talking at this point. I can show the wiki, for example, where we have Rob's model for the spike protein. Amanda, do we still have time or should I just continue?
Yeah, we have a few more minutes. I think people are trickling back in. So let's, go ahead, maybe just 3 more minutes or so, and then we'll.
Okay. So I'm going to go to a wiki page that we have. Right now, the wiki page is not directly linked from our production pages, but it's publicly available. It's wiki.glygen.org. And the reason we have not connected it is because it's still under development. We are figuring out the structures and so on. But there's already important and interesting information that we have. So when Rob came, we found out from Rob that he's working on the model, 3D model of the glycosylated spike protein. We got this model from him, and this is now available, the initial model that he produced, and we also have the overlay of snapshot from MG simulation of the S-glycoprotein from SARS-CoV-2, and this type of information can be quickly made available through these wiki pages. We are also planning on providing more FAQ and other pieces of information that can be connected to individual proteins. Mike, Renee, you want to add anything to that?
No, I think that's fine. I would like to thank everybody that joined us and provided us with, new ideas and potential use cases that we can integrate on the web page. But even, beyond the meeting, if you're interested in GlyGen, the web page is free. Just go there, play around with it. And if you have ideas, questions, concerns, just contact us. We are very responsive. We need your input to make that resource a success because we are developing it for the community. That means for you.
Thanks everybody for your input. It's very helpful.